Hello everyone. In this study, we try to isolate, identify and characterize the probiotic bacteria from honeybee gastrointestinal tract. As you might have heard that many researchers all around the world are trying to find new sources for probiotic and bacteria, and in this case, honeybees are one of the favorite sources. Microorganisms in the intestines of animals can benefit their hosts by helping digest food, detoxifying harmful molecules, providing essential nutrients, protecting against pathogens and parasites, and also improving and development and immunity. So the microbiota in the gastrointestinal tracts of all animals are one of the main and important parts of the biological beings. And in this case, as social insects, honeybees possesses a unique community of gut microbiota. And there are three main phyla that dominate the gut microbiota of honeybees, and they are Protobacteria, Actinobacteria, and Firmicutes. And some of them inside this genera are having potential for probiotic bacteria because they are lab or lactic acid bacteria. For materials and methods, we did sampling and bacterial isolation, determination of acid tolerance, determination of bile salt tolerance, DNA isolation, and 16S rDNA PCR amplification. Also 16S rDNA sequence analysis and identification of the isolates. Sampling and bacterial isolations. The B samples, the FFB genotype, Apis mellifera anatolica, samples were collected from Aegean Agricultural Research Institute, which is located in Izmir. The samples were stored in sterile falcon tubes containing 10 milliliter of a normal saline, and then immediately were taken to the laboratory. Then B samples were dissected separately on a sterile petri dish, and then entire gastrointestinal tract removed septically under a laminar flow. The gastrointestinal system were homogenized in a normal saline. For lactic acid bacteria isolation, gastrointestinal samples were pooled and smeared on MRS, agar medium by serial dilution, and then incubated at 37 centigrade degrees for 3 up to 4 days under anaerobic conditions. To obtain pure cultures, the colonies with different morphologies purified by streaking culture method. For primary screening, gram staining, spore formation, motility, nitrate reduction, and catalase tests were performed. Determination of acid and bile tolerance. In order to determine acid tolerance, MRS and M17 broth's pH were adjusted to 2.5 using one normal sterile hydrochloric acid to create an environment similar to gastric acidity conditions. Cultures activated twice in 18 hours in MRS and M17 broths. The bacterial cell pellets then obtained were suspended using 7 milliliter sterile saline in test tubes with a pH value of 2.5 in 10 milliliter. MRS was inoculated into broths at the rate of 1% and then incubated for 3 hours at 37 centigrade degrees. Subsequently, the viability of the cultures was followed by serial dilutions and incubation at 37 centigrade degrees for 48 to 72 hours. For determination of bile salt tolerance, 7 milliliter of MRS and M17 broths with 0.3% oxgal were inoculated with 1% of the isolates. Later, viable bacterial counts were done after 48 till 72 hours of incubation at 37 centigrade degrees. For 16S rDNA PCR amplification and sequence analysis, the DNA was extracted using a commercial kit, and the following primers, universal primers, were used for PCR amplification. And then after sequences, some of the softwares used for the editing the DNA sequence, and then the BLAST database, 16S rDNA database, was used to identify the strains, and in order to create the phylogenetic tree, the MEGA7 software was used. So results in primary screening, a total of 15 colonies with the characteristics of lactic acid bacteria were selected. Acid and bile tolerance viability values after 72 hours are given at table below. So 5 isolates designated as M2, M3, M4, and M11 and M13, which were tolerant to acid and bile salt, selected for molecular identification. You can see the results here: viability, colony forming unit per milliliter, pH 2.5, viability for bile salt tolerance. Well, 16S rDNA sequence analysis and identification. According to our BLAST homology results, the species with high homology to our isolates are given at the table below. As you can see that except for M2, which belongs to Bifidobacterium genus, the others, M13, M4, M3, and M11, belong to Lactobacillus genera. And also the phylogenetic tree, you can see at the figure below, which was constructed using MEGA7 software by neighbor joining method. Thank you so much for being with us.